Okay.
Hello and welcome to the final installment of the 2021 Tyson Summer Seminar Series. It's my great pleasure to introduce Dr. Janis Antonovics from the Department of Biology at the University of Virginia, joining us today from Scotland. As infectious disease has been on all of our minds during this COVID pandemic, we are fortunate to hear from one of the preeminent researchers of the ecology and evolution of infectious diseases in natural populations. Janis's primary concern is human infectious disease, but he typically uses plants as tractable, accessible models for investigating the transmission and dynamics of pathogens in nature. This includes a foundational set of work on sexually transmitted infections of plants, as well as long-term studies of plant disease metapopulation dynamics. He has also researched diseases in organisms as diverse as bumblebees, mice, and primates, and analyzes data on human diseases. His scholarship also extends beyond empirical and theoretical research to include the fascinating history of evolutionary biology and germ theory. His work has been funded by the NIH and NSF, as well as the Humboldt Foundation. Yet his tremendous impact on the fields of ecology and evolution is not only through his own research, but also through his incredible mentorship through the decades. The long list of students and postdocs that he has trained reads essentially as a directory of the current leaders in ecology and evolution. Some notable scientists who have worked with Janis as undergrads include, but are not limited to, Peter Kareva, May Berenbaum, Sonia Altizer, Britt Koskela. His past graduate students include people like Helen Alexander, Keith Clay, Ruth Shaw, Debbie Roach, Pete Thrall, Jim Beaver, Mike Gilchrist, Ann Pringle, and Amy Peterson. And finally, his postdocs have included people like Charles Nunn and Michael Hood. So really a fantastic lineages of ecology and evolution have come through his lab. Janis himself completed his thesis on speciation in plants on metal contaminated soils at the University of Wales with Tony Bradshaw. His first faculty position was at the University of Stirling in Scotland. And then he moved to Duke University in 1970 and has been at the University of Virginia since 1998. Janis was elected a Fellow of the Royal Society of London in 1988 and to the American Academy of Arts and Sciences in 1992. He's a 1991 Guggenheim Fellow and won the 1999 Sewell Wright Award. And it's now our honor to hear his talk entitled Intertwining History with Disease Biology, a mere cryptogamic powder. Thank you for joining us.
Oh, thank you, Rachel. Thank you for that very nice talk, nice introduction and talk, actually. We could probably go home now, I think. It's very nice to be here. It's a real honor to give the talk of the Tyson Field Station series. And I was especially pleased to give the talk because what I want to really cover today started in the field station. It started at Mountain Lake Biological Station. Before I get there, I want to just thank my close collaborator on this project, and that's Michael Hood, where we together got a grant from NSF called Opportunities for Public Understanding of Science, OPUS, which is an acronym, and acronyms are dangerous. And as soon as you get into history, you think of something like old people's useless science. But we do our best. And what I want to tell you about is some of the work that really was not really totally a product of this grant, but basically was a product of our history, our interest in the history of the disease that we're studying, namely anthismut disease. The work, as I said, started at Mountain Lake Biological Station. And it wasn't started by me, it was started actually by Helen Alexander, now at the University of Kansas. And Helen actually did her thesis on plantago, the same thing that Rachel works on, and on a disease in plantago. And then she went to Mountain Lake and became interested in the disease I want to tell you about, anthismut disease. Mountain Lake is a lovely place. If you get a chance to go, do go. It's got a lovely old lab. It's also got new labs and these lovely cabins where you can stay. and play volleyball and have summer camp for biologists, basically. If you go to Mountain Lake, it's a beautiful area. You may see people like Doug Taylor, and we have a long-term research in environmental biology grant counting this strange flower, this white flower that is all around the roadsides and the hillsides around Mountain Lake. That plant is actually white campion. Its Latin name is Silene latifolia, and it's attracted a lot of attention historically. One of the reasons is it has two sexes. It has females and males, where the sex determination is very similar to that in humans. There are separate sexes. So that's fascinating, and it has intrigued Doug, who you saw on that slide, has done a lot of work on it. But our interest in it was, wow, when you go there, you see that some plants are clearly diseased. The flowers are diseased with anther smut. So instead of producing yellow anthers, they produce these dark smutted anthers that are producing fungal spores. And the fungus is now known as Microbotrium silenis dioica. And I'll probably just call it Microbotrium. It's a gee whiz situation, because when, as I said, the plant is dioecious, but when a female becomes diseased, and here's a female on the left, and on the right is a diseased female, she's converted into a male-like organism. She's forced, quote, by the fungus to produce anthers, which then produce the fungal spores. So the flower looks hermaphrodite. So it's an amazing disease developmentally, but it's also a wonderful disease to do research on. And it's a wonderful disease because the symptoms are really easy to see. You don't have to do any PCR or any fancy techniques. It also has a huge effect. It sterilizes the host completely. So it's not just the anthers, but if we go back, I can go back. That button won't work, not to worry. We go back, then we can see that it sterilizes the ovary as well. It essentially makes the ovary tiny and semi-aborted. But it has little effect on mortality. So we can go out and count the number of diseased individuals and also knowing that they haven't been killed. They're still there. It's experimentally very tractable. It can be grown like yeast in the lab, and it occurs on multiple species worldwide. It's been identified explicitly on over 100 species. And the real interesting thing is it has no agricultural or medical importance whatsoever. So we're not sucked into the applied imperative of having to control the disease. We can work with it however we like. We have to get permits to move it around the country and even to bring it into the country from abroad. But other than that, we're not worried about spoiling agriculture or infecting humans. I should add that we can also identify the species from herbarium collections. And it came to, and I was looking at the maps and preparing for the talk, and I noticed that if you go to Missouri, which I couldn't remember where Missouri was, but it's here somewhere. And I think you're actually located in areas where there is disease on Silene virginica, and there is disease on Silene, anthosmut disease on Silene caroliniana. One of the nice things is you can actually go to herbarium collections and now on online resources like iNaturalist and obtain super data on disease distribution without having to do it ourselves. The life cycle is fairly simple. Basically, here's the diseased flower, spores. The spores germinate, they undergo they're diploid, or their spores are diploid. The funguses are heterokaryon inside the plant. It undergoes meiosis, then it produces a yeast-like stage with two mating types, which have to mate before infection occurs. I just wanted to mention that one of the real surprises in this whole system is that it turns out that the fungus itself, that Microbotrium itself, has sex chromosomes, even though it's a haploid. So it's actually shattering all our ideas about how sex chromosomes evolve. And there is super work being done in Paris by Tatiana Giraud on evolution of sex chromosomes in the fungus that we work on. As Rachel said, my main interest in anthesmut has been as a model for human disease. The interesting thing about pollinator transmitted diseases, and this is one, is that they're analogous to sexually transmitted diseases. They're transmitted during flowering, when the plant's reproducing, and also to vector transmitted diseases. And our interest has basically been that in such diseases, all the classical models don't work. Basically, there's no density is much less important. Social distancing doesn't work. Basically, if you're a vector pollinator transmitting, moving between flowers, you just fly further if the flowers are further apart. You don't fly as far if the flowers are closer together. So density isn't as important. If you're a sexually transmitted disease, well, most organisms find mates at huge distances. Also, in humans, we don't actually have more sexual partners when the density is higher. There might be more opportunities in cities, but generally that isn't the case. We usually have a single or a few partners. So whether we get diseased or not by sexually transmitted infection is relatively independent of density. But that's not what I want to talk about. I might come back to this. I will come back to this briefly in the end. What I want to talk about is the historical aspects and the fact that this disease turns out to be pretty amazing in terms of its historical impact. I want to start with a few giggles and confusions before I go on. First of all, we call it a smut. Well, yes, smut means it's crude and it's filthy and it's pornographic, but basically the word comes from the term schmutz, German, and even further back than that, meaning blackened or dirty, because the anthers produce these blackened, dirty symptoms. Its sexual connotations didn't come till much later. It appears early in the literature. But it's first mentioned in Samuel Pepys's diary. When he saw a play, I saw this new play and did not like it, it being very smutty. And if you look at, and anyway, we can also research the play. It's very interesting. The other thing I wanted to say about confusions is that this smut disease is including our anthesmut is called Astralago. Astralago is the name for smut diseases. Used in the past, it was used as a generic name for our anthesmut, even though the corn smut, which is Astralago, and the wheat smut shown here on the left, are now known to be in a completely different order from the one that we study, microbotany. Even though at one point they were given the same generic name. They're convergent in their life cycle, or they're retained an ancestral life cycle. So what I want to talk about is how anthesmut relates to the germ theory in Linnaeus. Then I want to get into talk about anthesmut and Darwin. All of this came as a surprise to us, by the way. And then I want to focus on Darwin's blind spot, and then finish with some perspectives. I want to start with the germ theory of disease, and I think it can be best summarized by this one slide. This is the germ theory of disease, or the history of the germ theory of disease. Contagious diseases have been known since antiquity. We talk about social distancing now, but people were social distancing millennia ago whenever an epidemic came in. They fled the town. The main doctors involved, like Galen, fled the plague. And there have been theories around of why things were infectious, what's going on. But this quotation from a famous Italian historian who's no longer alive, unfortunately, but all of his works are stunning to read. He says, "The history of medicine is the story of a fundamentally mistaken theoretical paradigm." And that was supposed to be a parenthesis in there, which meant, namely, the theory of humors and miasmas, which succeeded in dominating medical thought for an exceedingly long period of time. How and why a totally erroneous paradigm maintained for centuries its uncontested domination of medical science remains one of the most fascinating problems in European cultural history. So we don't know why it took so long with microscopes, with knowledge of small organisms. It took so long for the germ theory to really only get going in the late 1800s. It all starts because Linnaeus classified a white campion with anthosmut as a distinct species. And then, and this is what got us into the research, he changed his mind. Why would he change his mind? It all started when Linnaeus worked as, he got his degree in Holland, actually. He had to go to Holland to get his medical degree. And then he got a job working on one of the directors of the Netherlands East India Company, who made a fortune as a colonialist in India. And he had a wonderful garden, and he had Linnaeus employed as his doctor, as well as cataloguing his garden. And in cataloguing his garden, he produced this book called The Garden of Clifford, Hortus Cliffortianus. If we go in there, this is well known because this is the time that Linnaeus first used the floral system to classify plants. If we go in there, we see that he named a plant. Oops, I went too fast, but let's stay there. He named a plant Cucurbitalis floribus dioicus. He named a plant which otherwise was Cucurbitalis floribus dioicus as Cucurbitalis floribus hermaphroditus. He said it was a hermaphrodite with 5 styles. And then listen to his description. The marriage bed is snowy white, yet in the venereal act, all the sheets are stained with a dusty black powder. It's a giveaway. Basically, he was talking about a diseased Silene latifolia, not a true hermaphrodite, but a diseased plant. Then, in Species Plantarum, which is the time he first started to use the binomial system. So you see, he has Lignus floribus dioica. That's the binomial system. There, below, he has Cucurbitalis floribus hermaphroditus as a beta, which meant he treated it now as a variety and not as a separate species. Wow. Why did he change his mind? When and how? So what do we do? Oh, I have to say, this wasn't casual. I've got to add this in. It wasn't casual. He actually says it is superfluous to include diseased plants as varieties. In a publication somewhat later, but before 1753. And he mentions Astralega. He mentions the smut of barley and the smut of oats. So he knew about smut diseases. Also, he named other things, and this is one's for Rachel. He named other things as separate species, including Plantago, when it's diseased by Fusarium oxysporum. He called it Plantago rosea. So it was easy to make that mistake. Questions. What led Linnaeus to recognize that this hermaphrodite plant was diseased? And what role did Linnaeus play in the development of the disease theory? So we have to pinpoint when this happened. So we can make trips, and these trips are a lot of fun. So we can make a trip to London, to Piccadilly. And there in Piccadilly is the Linnaean Society, now under threat by Boris Johnson and his Tory government. They want to sell it off and make money by renting it out to McDonald's or to, no, not Gap, that's gone out of business. But anyway, it's a beautiful place. It's where the theory of natural selection was read as a paper, the Linnaean Society. And this is the library. The interesting part of the Linnaean Society is the crypt, or not the crypt. It's basically the strong room, the safe where they keep all of Linnaeus's books, collections, manuscripts. And there you can actually go in and get his herbarium specimens. And wow, we can find a hermaphrodite Silene latifolia. You can see clearly that it has anthers which are dark and smutted. And there is the ovary. It's a reduced ovary. And it's labeled hermaphrodite. It's 6 beta. It's labeled as it was in species plantarum. So the specimen is still there, but we don't know how it happened. But what you can also do, you can leaf through, well, you can't really, you have to be careful and hold books carefully with beads and gloves. You can leaf through Linnaeus's books and his drafts. And one thing he did, when he did his drafts, when he had books made, he had a lot of them printed with blank pages. There was the original, there's the blank page, so he could make notes and then prepare for a second edition. It turns out that when he was preparing for a second edition of the Flora of Sweden, bingo, we find he has crossed out Floribus hermaphroditus. Bad, bad, bad. He realized he'd made a mistake. And he did that in Ljungskile in Sweden in 1752. So he realized he'd goofed. So had he just realized he'd seen a diseased plant or not? The puzzling thing, and I refer you now to the citation we've published on this with Michael. If you want to find out the full story, go and read the paper. But basically, I think he was embarrassed. He didn't mention this again in any of his work. But one thing we discovered, and I discovered this because I'm not a historian or a Linnaean scholar, and this is familiar to Linnaean scholars, but it's amazing how many books on Linnaeus you read that don't mention it. Linnaeus was very aware of infectious disease. He was a doctor. And very early on in his career, in 1750, even before he published Species Plantarum, he was positing parallels between animal and human diseases. If only we thought about it, we would find that in nature, it is the smallest things that have the greatest effects. Perhaps invisible worms have carried the cattle plague among all the cattle of Europe. Perhaps smallpox, measles, dysentery, French disease, syphilis, even plague itself are caused by the smallest worms. Linnaeus, and again, this is generally unknown, published 186 theses under the names of his students. There's a lot of debate, but the general consensus is that these were basically works by Linnaeus, essentially voiced by students. That's what I like to do. I just like to tell my students what to do, and then they write it all down, and it's great, And so there are literally hundreds, 186 theses. A lot of the early ones, the earliest one is on the tapeworm. He gets the head and the tail of the tapeworm mixed up, but nevertheless, he was interested in tapeworms. And then he published, or his students published, Neander and then Roos, published 2 theses on infectious disease, one called Living Rashes and the other one called The Invisible World. And we've been really into these, basically in 2 ways. One, what about them? Well, the first one, Living Rashes, was translated and an account published of it only in 1995 in English. And it was by Margaret de Lacy, who's a historian, and Arthur Kane, a biologist, evolutionary biologist. Many of you may know Arthur Kane. He was involved, Kane and Shepherd, in the work on snails, different colored snails and the selective advantage of snails in woodland and grassland in England. And he became the professor of zoology at Liverpool. So it was published by, the translation was done by a biologist in 1995. I tried to get Mundum Invisibilum translated, and I eventually succeeded in 2016 with the help of Koos Kritzinger, who was a Latin scholar doing mostly religious studies of religious texts in Pretoria, South Africa. And this had a role to play because I had tried to find 5 people, I think 4 or 5, but it was 5, it may have been 4, to translate this for me or help me translate it. I have schoolboy Latin, so I knew it was important. And it was actually Martin Kemmler, someone who works on Anthesmut, who had moved to South Africa, and we were in the field. And I was telling him about this. He said, oh, I know, my colleague, Kus, maybe would like to do it. So this translation is one that I won't say I did it because he did the Latin translation, and I helped with the biology and the notes. And we published this. It's amazing that these 2 theses were dealing with infectious disease were not available to the English-speaking world till very, very recently. And what does Mundum Invisibilum say? Linnaeus says, we see only a few things of the divine works. Many things bigger or smaller than these remain hidden, like the fact that many things bigger remain hidden, which is true when you think of astronomy. And we must speculate and investigate whether diseases, for example, intermittent and exacerbating fevers, owe their origin to infinitely small animalcules. And this is so prescient, it's uncanny. Hence, and he finishes with this, hence the smallest animals perhaps cause greater destruction than the biggest, and they probably destroy even more than all wars. But I cannot proceed further with my conjectures. Another day, and the diligence of a longer lifetime will bring these and more things to light. We can ask, why did Linnaeus's work have so little impact? Well, I'm still working on this, and I need to get more information. But basically, first of all, analogies between plant and human disease systems were not taken seriously. Secondly, Linnaeus got sucked in by the implied imperative. What I mean by that is whenever someone asks me, I'm working on disease, they always say, well, are you going to cure it? No, we want to study it. We don't want to cure it. We don't want to be sucked in by the implied imperative. Linnaeus was obsessed with his keys to medicine. He wanted to classify not just diseases, but the medicines that those diseases could cure. And it was very occult. In fact, the Swedish scholars have deconstructed Linnaeus. They've basically said he was a medieval mystic, still living in the era when every disease would have a corresponding plant that cured it, and so on. The main reason, though, was that Mundus Invisibilis made an outrageous claim, namely that smut spores germinate to produce animals. And this was ridiculed. It was ridiculed in plays. It was ridiculed by Voltaire, the French playwright and writer and commentator. Spores, fungi producing animals, it's silly. It's silly. In fact, Linnaeus didn't quite believe it either. He heard about this from a chap called Otto von Munchausen in Germany. And he claimed that spores of smut germinate to produce animalcules. They weren't working with anthesmut, they were working with barley smut and smut of wheat and of some, maybe of oats, but it was mostly barley and wheat. And Linnaeus writes in a letter to Munchausen, I have put the black dust of Astilago in tepid water, where the dust driving out animalcules brought me the greatest joy, and my eyes cannot be sufficiently satisfied by this delightful spectacle. There is no doubt that the cause of contagious diseases becomes clearer through this. He was excited, but, and this is a long story that I could give a whole seminar on, because we researched this and traced this and went through correspondence. He was wrong. Linnaeus was wrong, but he was also right. He made the mistake for the wrong reason. It turns out that the smut-like symptoms can be caused by nematodes. And what he was probably looking at was nematode seed gall. which is a nematode that enters the stem, goes up the stem of the wheat plant, and then starts to reproduce inside the seed and causes a gall. In 1822, Bauer even showed that the nematodes could be in the same grain as the smut spores. So Linnaeus didn't have the systematics of smut quite right. It's ironic. He was ridiculed. In his later editions, the section on this was left out of his Systema Naturae. And he was right, after all. They could germinate and produce living things, but it wasn't from the smut spores. In fact, the first experiments on infectious disease transmission were carried about around that time by a French chap, Mathieu Tillet, and these are well known and well published, translated. And he did these super experiments, which are essentially split plot designs. He had basically different treatments in the columns, different treatments in the rows of his plots, and he applied things at different times. And he showed that only seeds blackened with dust from diseased grains could produce disease in the subsequent crop, in the subsequent generation. And what did he do? He compared it with the Nero diseases. The smut is not a spontaneous origin, and it doesn't occur here except through contagion. And he says, this is Tillet, not Linnaeus, in reading the work of the physician who I cite here, one will note an analogy between the disease of which he speaks and that of smutted wheat. But what is the disease he speaks of? Well, it's venereal disease. It's a sexually transmitted disease. It's a treatise on sexually transmitted diseases. So Tillet was aware of this too. So why none of this penetrated the world remains a mystery. I'm going to fast forward in the interest of time. As I said, I could go on and on there. Back. Antismut and Darwin. The correspondence of Darwin is now available online. And Michael Hood, my colleague, floating through the web, found, wow, Darwin looked at antismut in about 2000, this is about 2004, 5, 15, 16 years ago. We thought, wow, what's going on? It turns out it was on May the 18th in 1863 that Darwin received a letter about antismut. It says, formal, this is formal Victorian era. Miss Becker presents her compliments to Mr. Darwin and takes the liberty of sending him the enclosed flowers of a variety of Lychnis dioica, common in the woods here. It has bisexual flowers and a large, dark purple anthers, which give the plant a very handsome, striking, handsome appearance. And I agree. I actually tried to get this patented once so that you could keep flowers looking attractive and different. The same conspicuous anthers occur on flowers bearing stamens only, one of which is sent. Miss Becker, this is the sender, does not know whether such a variety as this would be interesting to Mr. Darwin. If not, she must apologize for having troubled him. And Darwin, we don't know the reply. Let's go back. Go back. Who was Lydia Becker? It turns out that Lydia Becker was a leading woman in the politics of the Victorian era and a leader in the women's suffrage movement. She was the secretary of the Manchester Suffrage Society. She edited the Women's Suffrage Journal till her death and wrote some things which I recommend every man and woman read on the study of science by women and on some supposed differences in the minds of men and women. She was active in the suffrage movement and obviously lampooned, cartooned, and everything. This is one of the kinder cartoons on the left. Then on the cartoon on the right shows that it's a cartoon about politics and the election and what a bad hand Spencer Churchill had been dealt in the election. And the two people, the only two women here, are Queen Victoria in the bad hand and Lydia Becker. A bad hand because she was vilified, or not vilified by some for her liberal politics and wanting to give the vote to women. We don't know what Charles Darwin wrote to Lydia Becker. We've chased up the family. The family have been super cooperative, been trying to find the letters. But Lydia Becker wrote again to Charles Darwin. I'm indeed grateful that you wish to investigate the structure and the history of ligness. And then she goes on that she'd done some more studies and says, I hope it is not asking too much, that you would be good enough to favor me the result of your investigation. We don't know Darwin's next letter. Oops, I'm going back. I did go back then. Oh, Darwin also drew Anthus moth. He was sufficiently interested. On the back of the letter from Lydia Becker, he actually drew the hermaphrodite, female, the male with black anthers, and the male with common anthers. But then the next letter is a downer. Sir, Lydia Becker says, I am of course much disappointed that it does not possess the interest you at first thought it might. But it has procured me the pleasure and honor of communication with you. I'm sorry if I've troubled you. I hope they will not do mischief by spreading amongst other plants with the insinuating parasite with which they are charged. Lydia Becker herself was upset. You can go to the women's library. And I went to the women's library and picked up her childhood notebook. You open it, and there in the back, scribbled, are her thoughts and her anguish. And one of the things she writes, I submitted a few of the flowers to the eminent naturalist, Mr. Charles Darwin, and he wrote to me that the supposed large anthers were a mere mass of cryptogamic powder, analogous to the smuts of wheat. She was crushed. But she wasn't broken. She actually went on and continued her correspondence with Darwin. And she published one of the first papers on plant biology by a woman. There were lots of papers on botany and as a common subject for ladies to enjoy, but she was the first woman to publish a scientific paper on plants. And she did this all before she started her work on the women's suffrage movement. And it was the fact that she moved from the countryside to Manchester. She could no longer study her plants. She could also not get into any of the societies in Manchester, learned societies, the literary and philosophical society. And she was miffed. And so she became active in the suffrage movement. What was Sir Darwin? Well, Darwin wasn't interested. He never, as far as I know, mentioned Lydia Becker or Anthus moth again, even though it was a stunning case of adaptation of a pathogen. He had a standing interest in plant breeding systems as pathogen changed the breeding system, and infectious disease influenced his life. Darwin never published on infectious disease or contagion, even though his life was greatly affected by it. It was Darwin's blind spot. It was his blind spot. And I'll go on now. I don't want to go on too much to emphasize how much of a blind spot it was. He was a normal dad. Bless him. He was concerned about his children, and many of them were sick. They're listed at the bottom. You know, I bet none of you have had so many kids ill with measles and diphtheria and scarlet fever. I have examined, he writes to Hooker, I have examined primroses and cowslips, and I find what I said is universal. I ought to be curious to know which plants produce seed. Etty has perspired and we're in good spirits, the more so as Doctor has looked a little grave these 2 days. Says it's a form of typhus, an ugly word. And then he goes on, has Lichtenaltia flowered at Kew? Here's a picture of Lichtenaltia. It almost never appears in any mention of disease, except it comes across in the variation in plants and animals under domestication. And this is sometimes quoted. I've seen this quoted in people writing about Darwin and medicine. A particle of smallpox matter, so minute as to be borne by the wind, must multiply itself 1,000-fold in a person thus inoculated, and so with the contagious matter of scarlet fever. What's the context? It's his theory of gemmules in inheritance. but I have further to assume that gemmules, in their undeveloped state, are capable of largely multiplying themselves, like independent organisms. So he was interested in his pangenesis theory rather than in disease. Henry Holland's family doctor. My dear Charles, he writes, this is Henry Holland, also doctor to Queen Victoria. Though I know that worms are often a cause of irregular symptoms in young children, yet I cannot readily suppose that at the age of 11, Ascarides, which is the tapeworm, should be capable of producing the effects you described to me. If the salt injection should not be of avail, one including castor oil might be used to clear the rectum of these. You feel very sorry for poor Horace at that point. Who was Henry Holland? He wrote a book which went into 3 editions called Medical Notes and Reflections. He argued that animalcules caused infectious diseases. He even cited Linnaeus. In fact, his arguments, which I won't go over in the interest of time, were almost the same as Linnaeus. And he concludes, it's likely that many diseases are caused by animalcules. Darwin's mentor at Cambridge was John Henslow. He was the person who persuaded him to go on the Beagle on his voyage. In 1941, he wrote a report on the diseases of wheat. It was all known. I have observed wheat infected by 5 species of fungi, by the ergot, by the little animalcule that produces ear cockle, the one I told you about, the nematode, and by wheat midge, by a little insect. So, and he also says, you know, if you just keep things clean, you can avoid the seeds becoming infected. The reason isn't totally clear, and we have to go further, but one of it is that Darwin hated medical school. In his and this is often mentioned. In Mr. Duncan's lectures on Materia Medica on a winter's morning in Edinburgh, and I know what a winter's morning in Edinburgh can be like, there's something fearful to remember, and the subject disgusted me. I also attended on 2 occasions the operating theatre in the hospital. I rushed away before they were completed. The 2 cases fairly haunted me for a long year. I'm going to skip the next slide. What's the perspective on this? Well, maybe I'll go back, I think, to be clearer. Turns out that Darwin had almost no impact on our thinking about disease, especially infectious disease. And that impact declined with time. This is the work of Zampieri, and he's been doing a lot of work on Darwinian medicine and evolutionary medicine, its history. And he shows that the influence of Darwin declined with time. In the 70s, 80s, 90s, there was very little mention of Darwinism in any medical journal. In Darwin's time, there was some interest. There's a fascinating book by Kenneth Millikan, who was a doctor, on the evolution of morbid germs, who he speculates that diseases could change very quickly. Bacteria had short life cycles, and these might be evolving very quickly. There are also technical problems. I just thought I'd show you Koch's autoclave. I was in Berlin I'm often in Berlin now, but I was there a while ago, and I went into the Koch Museum, Robert Koch Museum, the Koch's postulates, discoverer of the tuberculous bat bacillus. And there's his autoclave. Wow, I thought, yeah, it was pretty hard to do microbiology in those days. Perspective. I don't know what to say. Okay, let's do it very quickly. Science flourishes, as does endless. Anyone can do this. You should do this for your organism. Go to the Web of Science. Look at the number of citations to your organism. This is in 1970 and 2020. And they all increase exponentially. And this one does too. It's lovely, very exciting. The maximum number of citations in 5 years to Homo sapiens is over 1 million. Drosophila, you guys eat your hearts out, is only 26,000. But look at how many there are for Anthracis. The maximum in the 5-year period, the most recent one, is 63, which is pretty good. How about citations to disease evolution relative to citations to humans? Ah, it's flat, it's flat, it's flat, till about 1990. Then it blips up. I don't know, I haven't got, it takes a lot of work to do it for every year, but 5-year intervals, it's dead easy. Whoosh. There's the Darwinian hiatus. There's my new term. The Darwinian hiatus. And suddenly it increases. And it's super, because the NSF, NIH, produce a panel on ecology and evolution of infectious disease. In fact, I was involved in some of those discussions. And I remember bringing anthisma to the table once because I thought we need to decorate a table. So I brought in some diseased Salini caroliniana I was working on, and everyone was, of course, very amused and very curious. So we do fine. What are we doing now? I just want to finish this very quickly. We're actually back working, well, we've been working on it for 30 years, but basically, so we've had several grants now from the National Institute of Health to work on anthisma. And we're very interested in the evolution of multiple different transmission modes. Transmission doesn't stay still, it evolves. If we look at the genus Microbotryum, and this is work by the person that led me to the translation, the translator for Mundus Invisibilis, Martin Kemmler. He generated a phylogeny of the smut. And if we map transmission mode onto the phylogeny of the smut, we can see that anther-smut, the trait of evolution of transmission through the anthers has evolved independently several times. It's also back mutated to aerial transmission. Just like human diseases, they too have often switched between different modes. The papillomaviruses are notorious. This is a recent paper showing where the virus is present in the mouth, the skin, the vagina and mouth, skin and gut, vagina. So where the disease is presented influences its transmission mode. Well, we now haven't left Mountain Lake. We're still doing work at Mountain Lake, but for a while, and now we decided to go to even more glamorous grounds, to Italy, and study this species, Dianthus pavoneus, alpine carnation, where the individuals can have 40% disease. It's like the plague of Dianthus. And what we're doing is basically looking at transmission that goes through vectors, floral spore deposition, and to seedlings. It turns out we inoculate seedlings if you want to get disease. And our demographic studies here showed that there has to be a lot of seedling infection to sustain the disease. So this plant is actually using 2 transmission modes. And so we're doing experiments that I'm not going to talk about. This isn't a science seminar. This is an impression here I'm giving you. So we go out and we do experiments. We work with a carnation. So it grows, we can stick it in floral tubes. We can put it out at different densities. We can have dishes. We can have healthy flowers as targets for disease coming from diseased flowers, dishes for aerial transmission, and we can get data showing that the transmission, importance of the different transmission modes, aerial and floral, depends on density. So let me finish, because I think I want to leave time for questions. Summary. Anthismut, this mere cryptogamic powder, ties together very critical moments in the history of the germ theory. I haven't yet solved Carlo Cipolla's problem. I have no explanation for why it took into the late 1800s for the idea that organisms could transmit disease to take hold. The reason is that now and then, research fields build up self-referential constituencies. Gosh, I need a drink of water when I say that. Self-referential constituencies. Maybe I need a whiskey. If only Linnaeus, it's nearly 11 o'clock. If only Linnaeus had been listened to seriously, how many lives might have been saved? By the way, historians hate this kind of argument. They hate the the just, what if kinds of arguments. If only Becker had investigated if insect vectors could transmit disease. We had to wait till Walter Reed determined that mosquitoes could transmit yellow fever. And then malaria was realized. That happened to malaria a few years later. That wasn't until the late 1800s. And what if Darwin had written a book on infection and disease? I've left out some slides. One of the things I left out is, if you look at how often something like antibiotic resistance is cited in the journal Evolution, the evolution of antibiotic resistance, it's cited quite rarely. And you find that sexual selection, which Darwin studied and was interested in, is cited about 20 times as much in the journal Evolution. So evolutionary biologists have been seduced into following Darwin in much of their research program. Fortunately, we finish on a positive note. I think things are changing, and they're definitely changing because of COVID. Finally, the idea that pathogens can change and can evolve and can escape vaccines. You can have vaccine escape mutants. They've tried to hush that up for polio. Polio's had vaccine escape mutants over the world. They've tried to hush that up. But fortunately, we now know a lot more, and it's more open to the public. Let me finish with this. I cannot proceed further with my conjectures. Another day and the diligence of a longer lifetime will bring these and more things to light. Anyway, but I am very happy to take questions. Thank you.
Thank you so much for that fascinating and very timely overview of a lot of history that I think many of us have never heard before, even those of us who study infectious diseases and have looked into some of the history of what was known when. So we're waiting for questions to be added to the YouTube chat.
Excellent, thank you. I don't know what I should do with my screen, but I'll let you handle that. It's blank now. I don't know why quite. Okay. Oh, back. I left out some equations. I left out equations. It wasn't a science talk. Sure. This was old people. This was old people's useless science.
There is a question now that I'll ask, and I'll save mine for the next pause in the questions. So Dave Queller asks, how prescient or unique was Linnaeus in terms of thinking about microbes as causes of disease? Have you investigated whether other less well-known contemporaries speculated about the same thing?
Yes, there's a lovely couple of books actually by Margaret de Lacy, who goes through almost person by person on germ theory of disease. And there were others that were thinking about this at that time. The most famous one is obviously Storrow, who was this Italian physician who actually thought that seeds were he thought there were several causes of contagion. One was contagion at a distance, one was contagion by contact, and there was another one, I forget which, but basically he suggested that particles moved through the air. And this for some reason was poo-pooed by all scientists except one or two. And most of the people who wrote about it speculatively. And they didn't really marshal the arguments. Linnaeus was the first one to marshal the arguments. And then, unfortunately, to his sad demise, to try and experiment on it and do an experiment. But then there was Tillet, and Tillet was actually not the first. The first one was someone who showed that with onion diseases that were soil-borne, these were rot diseases. If you actually isolated the bulbs by trenches in the soil, then they would not get infected. So there were others, but Linnaeus was the first to really put forward a cogent, convincing argument.
So one thing that really caught my eye in your presentation was about this essentially co-infection in the same grain by a nematode and the smut. And so I'm wondering if that's, one of the first documented examples of somebody studying co-infection of a macroparasite and a microparasite.
I don't know. I mean, that's a very good question. I haven't thought about when did the concept of co-infection come in? I've also been trying to trace when the concept of parasitic castration came in, because obviously anthosmut's an example of parasitic castration. But again, Lydia Becker was a lost woman and wasn't cited in some of the animal literature on parasitic castration. But in terms of co-infection, that case it's a very interesting situation. And I personally actually don't know much about it. One of the problems is that there's not much smut around there. It's controlled, both by breeding and by seed treat. So that there's no smut around. There may be nematodes around, but I don't know anything about seed gall nematodes. I corresponded a bit with, there is someone at Virginia Tech who is working on it, actually. I did try and get some pictures once.
Wonderful. Great. So Joan Strassman asks, since you've given
hi, Joan.
Since you've given a great overview and historical talk, maybe you have some good advice for new grad students looking to choose problems. And she, like many of us, well, all of us, wishes you could have been here to visit us.
Advice about choosing problems. Yes, think of simple things, ask simple questions, obvious questions. Don't get into jargon right away. Don't get mystified. Keep your childhood there, and ask obvious questions. The other thing is, don't do history. I mean, history isn't going to help your research very much, but if it gives you energy, do it. Whatever gives you energy, whether it's playing the trombone or studying history, do it. But I don't think history is there to inform you about what your next experiment should be. On the other hand, I think it's very important to say, what don't we really know about? And one of the problems is that graduate school is learning what we don't know. Undergraduate school is learning what we know. The hard part is, how do you learn what we don't know? And I don't have an easy answer to that. And you get into Don Rumsfeld and, knowing what, anyway, I won't go into the Rumsfeld mantra, but what you know, the worst thing is you don't know what you don't know. But basically, the main thing is to be simple, stay simple. We mentioned Jim Beaver, one of my graduate students, about soil feedback. Well, that was the simplest experiment imaginable. And I still wish I'd done it. I just didn't think of it. He did. Asking whether, if you grow a plant in a pot, whether it makes the soil worse for itself. And it does. And that's the foundation of a huge area of ecology now. And Jim is cited way more than I am. It's amazing the impact he's had. And it was a simple question. So keep it simple. Don't get bogged down by equations and jargon. That's my advice.
That's great. That's fabulous advice. There's a few more questions that I'll
yes,
ask
please.
From the chat. But first, just to kind of continue some of this historical perspective and thinking about what we do and don't know. And I'm wondering if as you're kind of delving into history, if you're seeing a lot of parallels between this extended period of miasma theory and the current problems of some members of the public, not believing in coronavirus or thinking it's a hoax and not accepting current science. Is it a completely different problem, or do you see some parallels?
I see some parallels. I think that the parallels are basically that we kind of believe what we feel emotionally rather than what we know rationally. And it's very hard. It's very hard for the public to understand how science is done. One of the problems is science is a private matter. They can't get the journals, they're too expensive. It's a bit better now. But, you know, you couldn't get the journals without paying for them. You still can't. I still can't get a lot of the papers without paying for them. It's very true of medical journals. It's a closed shop, whereas religion is open. Everyone can get religion. It's free. The Bible's distributed. Science is not distributed. People don't know how science occurs. I had a chap ask me the other evening in a restaurant, how long does it take to make a discovery? And I said about 3 years, and we've got into COVID, so it's amazing how quickly COVID's happened. But they had this background in similar viruses, in flu viruses, in vaccines. But if you start a project, gosh, you got to write a proposal, you got to get it funded, then you got to get the people together, then you got to write it. So I said at least 3 years. I think that's 3 years is optimistic. So it's having the public get a much more realistic view of science and having science more accessible. And I think that's would be a very important thing. And having more journalists that know science rather than just spouting sensationalist crap about the next discovery.
Sounds like good advice. And I like you specifically emphasizing the need for the public to understand not only the outcome of scientific discoveries, but about the process of science.
Yeah.
What happens. So one of the scientific questions in the chat is asking, how commonly do different types of anther smuts induce the production of spores from female flowers? Do all anther smuts cause female flowers to become hermaphroditic?
It's a tautology, meaning that yes, you define an anther smut as something that forces the flowers, forces the anthers to produce spores. But the phylogeny I showed you very briefly as an impression was of one genus. And within that genus, some species will infect flowers, other species will infect anthers, be a true anther smut, whereas there are also species, and one that we're interested in now is Silene otites, where they destroy the whole flower. And actually, in the case of Silene otites, it looks like it's wind pollinated. And it's a reversion, it's an evolutionary reversion. So the fungus enters the plant and then it can manipulate it in many different ways. And in terms of looking at the evolution of just the anther smuts, each of those separate branches of that phylogeny, especially the one on the right, that is all closely related species, each of which is fairly specific to the host that it infects, and it won't infect other hosts. And sometimes when they do, they produce deformed flowers. So part of the specialization to a host is to do the manipulation of the flower in the right way, so it's still available to the pollinator to visit. Because if you try and move the infection to a species it doesn't normally infect, it can produce deformed or sometimes miniature, ugly flowers.
Okay, so it's getting quite late by you. Let's go with one last question here. Someone asking, what did scientific publication and information exchange look like at the time of Linnaeus and his contemporaries? And what are the limits in information exchange? Who had access to these academic circles and societies? May have delayed progress in understanding things like germ theory.
It was a huge problem. In other words, if you look at Linnaeus's correspondence, and you just come across it over and over again, he's asking for people to send him books. He wants to get hold of books. And getting books was really, getting information, finding out about things was really difficult. And you did it generally by correspondence. And Linnaeus, fortunately, kept all his correspondence, and it was very extensive. But he never cites with the work of Tillet. I don't know whether he knew about Tillet. He corresponded with people in France, Jussieu and with Aymon. And he corresponded with Aymon, who actually studied smut diseases in barley. And Aymon, in his later publication, is the first one who suggests that Linnaeus made a mistake about anthisma, that being that being hermaphrodite. So he was on the ball, and Linnaeus corresponded with him, and that was actually, that could again be another talk. A fascinating correspondence. And so yes, it was very hard to get literature. I mean, a concept of literature was someone sending you a book. The quotation I had from 1750 on Linnaeus and the cattle plague in Sweden, that was a German translation from a Swedish publication. During that period, there was a lot of intellectual excitement. It was called the Enlightenment. And there were a lot of commentary journals. And so there were commentary journals which did abstracts of other journals. So there was communication at that level, but you could easily see how someone would just totally miss something. But it's unlikely people missed Linnaeus because he did become celebrated.
Okay, so there's several comments in the chat. Thank you for coming, and people saying that they've enjoyed speaking with you in the past, and found people with fun memories of Mountain Lake.
Good.
And so we all wish you were here, but thank you for joining us from a distance. And
well, it's been a real pleasure, and thank you for allowing me to give this sort of rather off the wall. I would say off the wall, but off the regular track kind of talk.
It was wonderful. I think we all learned a lot from it.
I hope so. Thank you.
Okay.